In this video, we are going to discuss about mechanism of the DNA ligase reaction. The enzyme DNA ligase has the structure like this. The DNA ligase enzyme has an attached NH3 plus group. Here is the NH3 plus group. The first step of this reaction is adenylation of DNA ligase. During the first step, this DNA ligase generally reacts with an AMP. This is the structure of an AMP. This AMP generally comes from ATP. In case of AMP from ATP, the R group is PPI or inorganic pyrophosphate. Sometimes the AMP comes from NAD+. When the AMP comes from the NAD+, the R group is NMN. NMN stands for nicotinamide mononucleotide. This AMP has a ribose and an adenine. This AMP also has a phosphate group. During the first step of the reaction, this AMP from the ATP or NAD+ generally reacts with the DNA ligase enzyme. Due to this first step of reaction, an enzyme AMP complex is formed. Here is the enzyme AMP complex. Along with the enzyme AMP complex, PPI from ATP or NMN from the NAD+ is released. During the first step of the reaction, the AMP is transferred to the LYS residue or lysine residue of the enzyme. The enzyme AMP complex formed during the first step of the reaction now reacts with the nick in the DNA. This gap in the DNA is called as nick. This nick in the DNA has a 5' phosphate group. The AMP, which is formerly transferred to the LYS residue of the enzyme, is now transferred to the 5' phosphate group of the nick in the DNA. The second step of the reaction is also reversible in nature. It is also reversible in nature. The second step of the reaction produces two products. One is the DNA ligase enzyme itself. Here is the DNA ligase enzyme which is released. In the second product of the second step of the reaction, an AMP group is attached to the 5' phosphate group in the DNA. The 3' hydroxyl group generally attacks the phosphate group and displace the AMP. As a result, a phosphodiester bond is formed. The phosphodiester bond generally seals the nick. As a result, a sealed DNA is produced. The displaced AMP generally released from the reaction as an AMP molecule. This is the released AMP molecule. So, the third step of the reaction is the displacement of AMP and sealing of the nick. In the E. coli DNA ligase reaction, AMP is derived from NAD+. The DNA ligases isolated from some viral and eukaryotic sources use ATP rather than NAD+, and they release pyrophosphate rather than nicotinamide mononucleotide or NMN in the step 1. The third step of the DNA ligase reaction is also reversible in nature. The third step of the reaction uses DNA ligase enzyme. Please like, subscribe and share.